Hello, everyone. Welcome back. Thank you again for joining us for live session number 3 for the Blue Cloud Hackathon 2025. We're just waiting another minute for more people as people are still joining us. But in the meantime, we can take a look at what we did last week. So we had live session 1, where we heard quite inspirational. We had quite an inspirational presentation on one of our jury members on why the ocean matters and why these challenges are the ones that we chose, and we think they are challenges that we should all be working on together to solve. Then we had another great session. End of last week, we learned all about the Blue Cloud V Labs, how to access them, how to work with them. And our presenters also explained a little bit about which V Labs can be applied to which challenges. So now we are just a few days away from the hackathon kicking off. And we have another really great session lined up for today. We have a lot of speakers. We're going to explore other features of the Blue Cloud 2026 services. We're going to take a look at the workbenches. We're going to take a look at the DD&AS, the data discovery and access service, as well as Beacon. These presentations are all going to be given by the team, by the mentor and coach team that is going to be supporting you throughout the days that you're going to be hacking. So again, this is a great opportunity for you to get to know them a little bit better, to take a look at their profiles also on the platform, and connect to them as soon as possible. It's going to be to your benefit. So we're going to start with some house rules. I'm also joined by Michele Erba, our colleague from Pristine Agency that a lot of you are already in contact with. And let's go through the house rules before we start. As you know, this is a session that is being recorded. We make these videos afterwards available on the platform, on Discord, etc., to be used by you as resources. They have some really good presentations in them, as well as showing you how the platform, the Blue Cloud platform works live. If you have any questions, please submit them through the Q&A. The chat is not going to be functional for this session. We are going to try and answer all of them live. And if we do not manage to get to them, you can always put your questions also, of course, on Discord, where someone will get to you as soon as possible. Yes, and then as usually, we're going to try and finish by 5 p.m. Again, we do have a lot of presentations today, so we'll see how that goes, but we're always trying to respect the agenda. So we're going to jump right into it. We're going to start with Nydia, Catalina, Reyes, Suarez, and Megan Ann French, both working for OGS. And they're going to give us a presentation on the EU of Workbench for Eutrophication. Nydia and Megan, thank you for joining us. If you can open.
Yes,
Hi. Hello, everyone. So I'll just share
my name
screen
is
now.
Megan French.
Okay. It's
Great.
It's my
Go
screen
to the next one.
being shared.
Yeah,
yes, it is.
Yeah, that's fine. To the next one. Okay. Yeah. Hello, everybody. My name is Megan French, and I'm presenting with Catalina on Blue Cloud. First, what is eutrophication? Eutrophication refers to nutrient over-enrichment, usually due to inputs from agriculture and urban drainage. It's defined within the Urban Wastewater Treatment Directive as the enrichment of water by nutrients, especially compounds of nitrogen and phosphorus, causing an accelerated use and undesirable disturbance to the balance of organisms present in the water and to the quality of the water concerned. As illustrated in the infographic, eutrophication leads to increased plant algal biomass and may lead to loss of biodiversity, ecosystem degradation, harmful algal blooms, and oxygen deficiency in bottom waters. Next slide. EMNET is the acronym for the European Marine Observation and Data Network, which is the European Commission's in situ marine data service of DG Mare. As one theme of EMNET, EMNET Chemistry publishes eutrophication data collections with parameters relevant to Descriptor 5 of the Marine Strategy Framework Directive, which states that dissolved oxygen and phosphorus are the main inorganic nutrients in the water column responsible for the eutrophication of marine waters, and that EU member states need to look at the levels of nutrients in algal blooms, the transparency of the water column, and the levels of dissolved oxygen. Now I will hand over to Catalina, who will talk about the workbench.
Okay, so for the Google Cloud project, the Eutrophication Workbench was set up in order to, as Megan said, not only EMNET gathers all this information about eutrophication, but there are other blue data infrastructures, mainly Copernicus Marine Service and the national, the NOAA, National Oceanographic Administration of the US. So the idea was to be able to merge these BDIs in order to find and produce the best harmonized and standardized collection of data, in situ data for eutrophication to evaluate the good health of our oceans. And so the first phase was trying to develop this, and the concept and the idea of bringing Beacon, which my colleague Robin will talk about later, was used to in order to merge these data sets, but not only merge them, but to create a set of rules in which these will be harmonized and standardized. Then the workbench will test and validate these data sets and document. And then at the end, we will have a set of harmonized, validated, and validated data collections for a couple of EOVs or essential ocean variables in chemistry, mainly nutrients, chlorophyll, and oxygen. So currently, only monolithic instances of Beacon are available for CMEMS, EMNET, and the World Ocean Database. And they will be available to explore in the hackathon. The merge instance will be released by the end of the project, and so please stay tuned. This is the team, the Eutrophication Workbench team. It's a team of European experts, and we all work close together to try to get this product in the best way possible. Okay, now I'll go directly to the live demo. Unfortunately, I have some problems now running the WebODV, but we'll see if they are solved by now. And so the first thing you have to do is to go and grab the Jupyter notebook that lives within this GitHub link. And so this link will point to this notebook that you are going to download, copy, and then bring into your Blue Cloud Lab. You will run here Blue Cloud Lab. Then you will open a Jupyter Lab. And in the Jupyter Lab, you will be asked to choose your server. And here you can walk around your workspace, create folders. In my case, I just created this Eutrophication Workbench folder. I copy paste my EMNET Chemistry Hackathon notebook, and the first thing you have to do is to install the Beacon API. Then Robin will go through this more in depth, and you will call the API. So you import these two libraries. Then you will need to use a token and a server. In this case, I'm using the Beacon that lives in the Maris server, but you can also use the one in D4Science. They are copies, they are mirrors, so it should be the same. Then you run a couple of lines And in this case, this line, what it tells you is what kind of data you can find there, what kind of information, not only data, but also metadata. So for example, in DD&AS chemistry, you can find the cruise, the station type, long lat, Atmo code, instrument information, all these, and plus the variables. As my colleague Megan said, for eutrophication, we are looking for signatures mainly for a couple of parameters. So that's it, dissolved oxygen, phosphate, chlorophyll, and nitrogen. So for example, in this case, if you want to check if within this beacon instance you have chlorophyll, you just need to type here CHL or chlorophyll, and it will tell you the chlorophyll species that we can find. For this little demo, I will just use chlorophyll A. I queried dissolved inorganic nitrogen, DIN, nitrate, and oxygen concentration, what the body saw of oxygen concentration, and phosphate. One important part that probably you know, but if you don't know, all this data have been quality controlled. And so it's important that you not only get the data, but the quality flags that are attached to this data. So you can know if there are some problems or if they are good data or not. I selected the range, the last 4 years from 2020 to 2023 December for the full domain, in this case, all European seas, for the depth from 0 to 100 meters. This because the eutrophication signal is said to be stronger in the first 100 meters of the water column. You can also create some filters, like long lat filters. You just need to add the polygon area. Then you query this data, you ask Beacon to retrieve that information, or you will find a data frame with the data containing what you asked. We created a little bit of statistics. So in this case, for example, we will see the mean, the standard deviation, the minimum, the 25th percentile, 50th and 75th percentile, the maximum of each parameter. Keep in mind that here all quality flags are taken. So high values could still exist here so you have to be careful. Then the availability percentage per parameter. So as you can see, for these 4 years, we have 0.4% of DIN, 3.9% of nitrate, 47% of chlorophyll A, 79% of dissolved oxygen concentration, and 3% of phosphate. So there is not a lot of data, but this is the data we have to work with, since it is very important data, since it is in situ data. Here what we did was just to plot the amount of data available per parameter per year. So you can also check, this is what I did, but you can also use whatever you want to do the statistics. And so in this case, in the year 2020, you can see how much of data you have for all the collection. So usually you have more oxygen and not so much nitrogen. Then another thing we did was to have to check what is the availability per depth range, and so this is the statistics per depth range. So in the first 10, 20, 30, 40, 50, 75, and 100 meters of the water column. And here in this block, what you're going to do is to select what you already did. It's a faster way to query Beacon and to download the data. So in my case here, what I'm going to do is to select these 4 metadata that for me makes sense to then find more information about the data I just queried. And so for that reason, in this case, I will just download the ODV. So you can have this drop-down menu where you can select if you want an ODV, NetCDF, Parquet, or SAR file. I'll download the ODV, and then I will show you. And my colleague Sebastian will show you more in deep an example. And then you just download this, and it will be saved in the folder that you were working in. So as you can see, we have the zip file here. This zip file, you will unzip it using your terminal, and then what you're going to do is to use WebDV, which I tried to start, but it's taking a little bit. Maybe I just need to close this. Okay. So in my data space, I should be able to see the collections I prepared. They are not showing. I hope that we will fix this by next week when we start the hackathon. So we will work with Sebastian on how to. But what you will see here is the data space, and what you have to do, and Sebastian will show you later, is how to import those spreadsheet files, which you get. Oh, anyway, yeah, now I'm at what you get when you use your notebook, and I can show you here. You will have 4 files. One divided by data type, so profile, time series, and trajectories. Errors are data that do not fit within these 3 categories, but rarely you have some of those. That happens when you have the merged collection. So in this case, for example, for the embedded chemistry, you will not find errors. And so I will show you quickly with how it works with the ODV desktop version. So what you usually would do with ODV would be to drop in the file and wait a moment until it loads the data. So as you can see, these are all the data that we queried for those 4 years. It can take a little while.
Catalina, we cannot see ODV.
You cannot see?
No, we only see the browser.
Oh. Thanks, Sebastian. Can you see it now?
No, it's the same screen. You might
It's the
need
same screen.
now, maybe.
Is it now?
Now, yes.
Yes.
Okay. So in this case, we have the stations have been loaded. And ODV is a great tool. Many oceanographers use it because it makes our life easier. So here what I'm going to do is to look at the full domain. You can go through the ODV how-to get all the functionalities. What I want to show here is that ODV allows you to check quickly the quality flag of the sample that you are looking at, and it can allow you to do some quick plots. For example, also here I wanted to show you the platform. We can look at the unique values, and all this data have a platform type. So we can see that this collection is joined of drifters, fishing vessel campaigns, maybe you have ship opportunities, all these. Okay, you can explore all the metadata. You can also query more metadata and get more information. With the CDI RD, you can get more information. So you can use this code to actually go in the CDataNet portal and look for the CDI, and you will have more information about the metadata and the provenance of the data. Then here we can do some statistics, like time Instagram. I wanted to show you very quickly because I know time is running out, but unfortunately I prepared what is it? No, sorry. Views. variables. Okay. So what I'm adding here is just to see the available data in the first. And I wanted to do layout. Sorry. No, I cannot do the views. I'll do surface. Okay, so here we can see the data availability. And example, this is oxygen. We don't have chlorophyll. I mean, this is all the things that you can do many things with ODV. I'm just showing a couple of things. This is chlorophyll, nitrate, oxygen, DIN. As you saw, we have very little DIN, and phosphate to show phosphate. Okay, another thing we can do is to get some other layouts such as the scatter, and in this case, for example, we can plot the data and see the quality of the data. If you check, for example, the display style and you choose the X quality, for example, in this case for nitrate, you can see some of colors. One is it's number one, which is good data, and red, which in this case would be 3 or 4, it's probably bad or probably or bad data. So you can play around with these collections. You can download the data, check the quality, make some statistics, make some further analysis, and yeah, that's it for me. I hope it didn't take long. Thank you.
Thank you so much. It was such an interesting presentation, and definitely will be super useful again as a resource. So I believe it was worth it. Thank you both, Megan and Catalina. And now we're going to quickly go to our colleague, Simona Simoncelli. Hi, Simona. Simona is going to speak to us about the E of Workbench for physics. So
do you see my screen?
We see your screen.
In
we see all
presentation mode?
There, absolutely. Yes, this is it. Yes, great.
Okay, so good afternoon, everybody. My name is Simona Simoncelli from INGV. I'm leading the EOV Workbench for Physics in the Blue Cloud project, and together with my partners from Ephemer, POC, OceanScope, and HCMR, we are working on a pipeline to build a pipeline and a workflow for ocean physics data, in particular temperature and salinity, and we do it in collaboration with Matthias, the National Oceanographic Data Center, and the RV Institute. So let's start from an overview of what is the Blue Cloud Open Science platform. So we have actually a virtual environment, which is at the center of the slide, that interacts with the Federation of Blue Data Infrastructure. So we are capable to gather data, access data, from this Blue Data infrastructure, and this data can be used on the VRE to build and to run application on virtual laboratories, or we can use them in workbenches to do data cleaning, data processing, and deployment. So
Simona,
this,
sorry to interrupt you. We can see not in the full presentation mode, so we see
okay,
a part of the screen with many slides.
Okay.
So yeah, maybe that could be it. Maybe slideshow. Use slideshow.
This?
Yes, there we go. Yes, perfect. Thank you so much.
Sorry.
All right, no problem.
I have two screens, and I was actually looking at the main one, and
No
it was
problem.
correct. So actually, where the concept of the workbench came from is the value chain, data value chain. So if we start from an observing system, a global observing system, we can get a lot of data, and this data can be ingested in blue data infrastructure. What is the, let's say, vision of the Ocean Decade from UNESCO is to build up an ecosystem of data infrastructure. And from this infrastructure, actually, the aim of Blue Cloud is to gather data from different sources, and through the Workbench's workflow, to integrate this data coming from different sources to actually compute indicators, derive report assessment, which can be useful for early warning systems, decision making, prior planning. And so the Workbench design is this. So we started from the idea to gather data and metadata from different infrastructure, and in particular from the Copernicus Marine Service, from the Sea Data Net, from EuroArgo, and the World Ocean Database for the physics. And so in our cloud environment that you see depicted here, we want to have a data lake from which to gather data and metadata. We want to harmonize the data to be able to integrate the data and identify and remove the duplicates, and run additional QC in order to let's say, provide to the user consistent data in terms of quality. So the output of the Workbench is going to be the added value data sets for temperature and salinity, but also the operational workflow that we are building. The idea is to test it in the Mediterranean domain, but we want to go global. So what it is, a cloud-based workflow is a series of processing tools that can be deployed on a virtual research environment and can be chained to complete a task. So the automation and management process can increase the efficiency and replicability of the process that we are implementing. And in this way, we can also optimize the quality of the final outcome. The implementation of these operational workflows rely on the FAIR principle. So data, services, all the digital assets should be findable, accessible, interoperable, and reusable. And the advanced services for that access, web app, for software development and interactive computing are the ingredient of this new approach to scientific process. Of course, this new approach is collaborative, and the Workbench example is, let's say, one of the example of this collaborative environment. So this is another picture of the cloud-based environment that can actually drive to what is now conceived as a digital twin. So again, we start from the blue data infrastructure that we have, and we can ingest the data to a data lake that can be accessed in the virtual environment to actually build up workflows that can also include models and AI application. And the idea is to derive fit-for-purpose products to the users. This product can be data sets, model output, and also what-if scenarios. So what we have built for you for the hackathon from the Workbench physics is this diagram. So we have prepared different notebooks that allows you to gather data from 4 different sources: the Copernicus Marine Service, the Sea Data Net infrastructure, the Argo, and the World Ocean Database. You have Jupyter notebooks that have been prepared by the expert, and these are giving you output files in different formats. In particularly, if you want to explore the data, visualize and duplicate, check duplicates and do quality control, what you can do is to use the ODB output format. And what you can do is to import the data in WebODB directly on the VRE and explore the data. So this is actually a diagram, again, that is explaining you how the Workbench team has set up this notebook for the rapid and easy access of these data sources. This means that the expertise from the data expert, the knowledge of the data expert has been blended with the knowledge of the software developers and for code optimization. So these notebooks that you find on the GitHub repository are having subsetting capability, filtering, aggregation of parameters, and conversion of different units. And the output, as I said, can be in ODB, which allows you to import the data and explore the data directly on the VRE without downloading the data. Or you can also select another Argo format optimized for the cloud environment in order to speed up your proposed project application. So here you have two screenshots. One on the left is showing you the notebooks repository in GitHub. You have the link here. And you can find one notebook for each single source. And on the other side, you have a screenshot of the environment, actually of the physics Workbench lab that we used, as I said, in a collaborative approach with the team members to set up all the different notebooks. So what is actually, this is an example from Cora. Cora is the Copernicus product for historical data. And in this notebook, what you can do is actually to select per platform. You can gather, well, you can actually retrieve essential metadata like platform, instrument, the data provider. You have subsetting capabilities because you can select the time period, the box domain, but you can also design and a polygon that can, for example, skip some regions that are, as a topography, very complex. You can select a depth layer and the variable you are interested, but also the relative quality flag. What you do is pressure to depth conversion in order to have all depth values. You can aggregate this notebook aggregate parameters. That means that some variables have different nomenclature in these different VDIs, and everything that is related to temperature is aggregated in one unique variable. You have filtering capabilities because you can, for example, select the data sets, like from the World Ocean database. You can select if you want to look at gliders or moorings, or if you want to select Argo profiling floats, or you can decide to keep the entire data collection. You can select the quality flag, as also Catalina was mentioning. So in these notebooks, actually, we have selected only the good quality data, but to facilitate your application. So you, if you're using this filter, you're gonna gather only the good quality data. And you have multi-output, pandas, GeoPandas, NetCF, ZAR, Parquet, GeoParquet, ODB, Arrow, and CVS. So let's have an example. So if you, let's assume we want to retrieve all Argo temperature and salinity values in the Mediterranean Sea between 0 and 150 meters in the time period 2020-14, only filtering good data. And get a Parquet output file. So this is actually what you can do with the Argo Jupyter notebook. You have here a screenshot of what is the table of number, an example, let's say an extract of what it is, the table of data that you are actually retrieving for your inspection. But we have also included some plotting script, like some cells in the notebook that allows you immediately to visualize your data. There are some statistics by year, so you can see the data distribution in time, but we have also scatter plots for temperature, salinity, but also temp TS diagrams to look at the water masses, for example, and the different layers that you want to analyze. And this is in Parquet, but in the same time, with the same Jupyter notebook, you can also run it with another option to get the ODB output. And what you get is a zip file. So what you have to do is to copy your file and unzip your file. And what you get, as Catalina was saying, unzipping your file, you get 4 different TXT, the errors, profiles, time series, and trajectory. What we suggest, for example, if you are interested in profiles, is to import this TXT file. You're gonna see Sebastian's presentation for more details. So what I'm going to just give you a quick overview. In OGV, so you are able to go to the same folder where you have unzipped your file and you can import your file. You can then, once the import is finished, you can be able to explore this data because you see it already in your data space as an ODB collection. So this is how you're gonna see it. So you're gonna see the web ODB interface, which is very similar to the ODB desktop that you might already know. And what you can do is exactly mimic what you can do in your notebook, looking at the time distribution, scatter plot for temperature and salinity. And, you know, you are planning of ODB user guide or example in the literature on how you can visualize and analyze your data. So just to summarize what you can do with the Workbench physics notebooks is to gather data in any region that you are interested in. And what you can do, like for example, here we did it for the Mediterranean. And what you can do is easily look which of the different BDIs is giving you more data or better quality data. The question, so at the end, is what is the data set that is fitting your needs in terms of core coverage, quality. So yeah, this is, I'm happy that you're gonna try it, and please provide feedback because for us it's very useful to tune these tools to your needs. Thank you very much.
Simona, thank you also for pointing out a couple of things. I mean, besides the great presentation on the physics notebooks, etc. You mentioned also the FAIR principles, which we have also brought up recently, and they're also part of the deliverables. And generally, as you can, and also the feedback that indeed we are quite looking forward to for the Blue Cloud 2026 project. And a lot of these presentations, as you can tell, are they're very useful to see one after the other. So nothing is everything is interrelated. In any case, thank you. And now we're going to pass to the E of Workbench for Ecosystems and our colleagues Jean-Olivier Irisson, who is being supported by Virginie Sonnet, Victoria Bancel from the Sorbonne University. as well as Meike Vogt, Alexandre Schickele, Corentin Clerk, Matthias Münnich, also Hofmann from ETHC. Wow. Okay, Olivier, over to you, Jean-Olivier. Thank you.
Yeah, that's a lot of people. So I guess you can see my screen and the slides are working.
Yes.
So first, some pictures behind the names and the roles of all these people. So Meike is the leader of this workbench. Actually, Lionel, myself, and Matthias were all researchers together with Meike, and we contribute to the supervision of all this. The Alexandre on the right in the ETHZ team is the one who has authored most of the code of the tools that are in the workbench. Corentin has contributed to this. Virginie on our side now is a user of those tools and has gained expertise in using and modifying them. And finally, Urs and Victoria have contributed to making them run as smoothly as possible on the Blue Cloud infrastructure. So it's really a team effort. And the goal of this workbench is to gather ecological data and extrapolate it. So that comes from a few points and extrapolate it to the whole world. And we're particularly specialized on plankton. And so to explain all this to you, there was actually a video made, which I'm going to play now.
Our Earth is a blue planet. When we imagine ocean life, we often think of whales, dolphins, or coral reefs. Yet the foundation of marine ecosystems is largely invisible. Marine plankton, microscopic organisms drifting with the ocean currents, forms the basis of the food web. Crucial players of the complex marine biological carbon pump, they contribute to the storage of carbon in the deep ocean. In doing so, they quietly help to regulate Earth's climate. To study these vital organisms, researchers around the world rely on a rapidly growing array of technologies. Seawater samples are collected, microscopic imagery is analyzed, genetic material is sequenced, and environmental conditions are recorded across a diverse marine regions. These data come in different formats, resolutions, and collection methods, each introducing its own biases, uncertainties, and gaps. This creates inconsistencies that make global synthesis and comparison difficult. At the same time, if we take a step back, we are facing a global scarcity of data, leaving us with a fragmented view of the ocean ecosystem. Our automated pipeline, Cephalopod, aims to integrate and standardize this wealth of data by connecting seamlessly with global data initiatives such as Atlant-Eco, OBIS, and GBIF. Through an ensemble of modeling algorithms and machine learning, Cephalopod enables reliable prediction of plankton distribution across space and time, intercomparable across all data types. Its high degree of automation and integration across projects and disciplines foster collaboration. Ultimately, Cephalopod is more than a modeling tool. It is a crucial step toward globally coordinated science-based stewardship of marine ecosystems.
So as you see, Cephalopod is really the central tool developed by the workbench. It's a way, again, to extrapolate from a few points to the whole world using the environmental data. And there's two ways to use it that you will be able to use in the hackathon. The first one is through the VRE that would be set up for the hackathon. And so this will be explained by Alexandre.
Hi there, Alex for the presentation for the ecosystem workbench for the Blue Cloud Hackathon. Welcome to the RStudio VRE image, and I'm going to present you today Cephalopods, which is a habitat modeling tool used to map the global distribution of biological targets according to environmental conditions in which we observe them. So I'm here in the RStudio session. The codes and documentation are here. So home, Blue Cloud Data Space, Ecosystem Workbench Hackathon. Please take this zip file and unzip it in your home directory. So there it is. There it is. You have all the README that are here. I don't have time in 5 minutes to go through everything, so please read the documentation here. There is an extensive explanation of all the options, how to use it, tutorial, input files, and so on. The main thing you will interact with is the master script here. It starts by clearing up your R session, loading all the packages, and defining your run name. So we start here by listing all the data that are available here in the example inputs, which is a CSV file. It is also used as a template if you want to use other biological target from maybe other VREs or VLabs. Otherwise, you can also source occurrence abundance biomass and metagenome assembled genome data from online databases. Here is a little custom block of codes where you define a vector of character, which are identifiers of species that you want to model. So here, for instance, this number is the worm's ID for zooplankton species Calanus finmarchicus that we run in the example here. And then we define all the parameters of the model. So this is set by default. Again, sorry for the time, but in 5 minutes, it's really hard to explain everything. The main thing here is that you define your data types. So in this example, we work with presence only, but we can also work with continuous and proportion. So it's basically the model is a wrapper and will go to a different algorithm depending on the data set or the data type you are working with. Here you define environmental variables. There is a set of predefined variables in data features. If you want to use others, please check the template. And the rest is fully automatized. So querying the biological data, matching it with the environment, generating pseudo-absence if necessary, checking all the inputs. So at this stage, we should have biological inputs that have no outliers and environmental inputs that are meaningful, likely to explain some variance in the biological targets, not correlated together, and not redundant. So basically a parsimonious set of environmental features. And then we do the modeling, which is an ensemble model approach. So generating the splits, defining hyperparameters, fitting the model, evaluation, projection, and then we have the outputs. Everything you do here is saved in a query object that is quite extensive here, or in a model object. So you can get everything you need. All the outputs are in these objects, and you have one of these objects per species. So if you go and look in the outputs, here I have my species. If I have several species, I will have several folders in there. Call is all my options for traceability. I have a log file here. Log files again for all the species. Here I have my query and model outputs, and I also have a whole bunch of PDFs. And I will go to the last one directly. So standard maps here. That's our output. We have here a traffic light quality check. Green light is satisfying for these models. This model didn't pass the projection uncertainty quality check. And here you have the projection. That's the input data, so where the species is present. So that's our presence only records. And here uncertainty, green is standard deviation, red is extrapolation outside the conditions of the samples. So as you can see, it's a northern hemisphere temperate species. So obviously in the Southern Ocean, we are completely extrapolating and we cannot evaluate the model there. And then you have an ensemble model as well with the quality checks, right? If you have any question, please check out the description in the PDF files. Check out Victoria's video just after it if you want to model more than a few species. She will explain how the CCP works, which is a more performance method, but non-interactive. And see you at the hackathon.
Thank you very much. Thank you. Jean-Olivier, anything else?
Yes, yes. So as introduced by Alex just now, There's another way to use the same method, which is through the CCP. So I think the CCP was not explicitly presented so far. So it's a way to use, so it's for cloud computing platform, and it's the way to use the power of cloud computing, which is not only having a VRA, but possibly running the same code on many nodes. So as long as you model, let's say, up to a small dozen, or maybe I think the maximum was actually 8 species, it can fit on a VRE. If you want to model hundreds, because you want to study diversity, for example, that won't be possible on a VRE, and probably, I mean, certainly not in the space of the time of the hackathon.
Mhm.
So the solution for this is the CCP, and Victoria will explain in another, but last video, how that works.
Thank you.
Hi everyone, I will show you how to run cephalopod pipeline using the cloud computing platform of Blue Cloud, which is an environment designed to execute containerized methods. So here I'm in Analytics Engine tab in the cloud computing platform. I choose the Hackathon cephalopod with parallelization method. So here you have to provide few inputs. Runtime inputs is a Docker image used for the execution of the method. So you don't need to change it. You can't change it. For the predictors, you have to provide a path in your workspace to your environmental data. So a zip file. And you have to choose protected link because in the code, we use a token to access those data. Then you have to provide a run name for your execution. So for instance, cephalopod run. Then again, you have to provide a path in your workspace for a custom CSV file which correspond to your biological data. Same, you have to choose protected link. Then, depending of the data in your CSV file, you have to choose a data type for instance, presence only, continuous, or proportion data. You can provide also species rank if you want to filter your species list depending on a taxon rank. Here I just put none. And here it's a key difference with running cephalopod on the VLab is that you can specify a number of replica on which the pipeline will be parallelized on. For example, if you choose 2 replica, the species will be divided into 2 batches with approximately the same number of occurrence. And the pipeline will run in parallel on both groups. So make sure that the number of species in your CSV file is greater or equal to the number of replica you select. Finally, you select the output path in your workspace. At the end of the execution, the zip file with the output will be located here. So same, you have to choose a protected link. Then you have, you can also choose which output you want to archive at the end of the execution. So if you want to archive the whole execution or compressed outputs, or if you don't want to archive anything automatically. If you choose the 2 first option, you will be able to find your output under CCP execution in your workspace. But in any case, you will find your output in the path you provide here. Then you can also optionally choose to provide at the end of the execution standard output to see the messages and the warnings of the executions. Then when everything is set, you can simply click on execute. And you will be able to find the live status of your execution. So if it's running, after you just execute, you will have your method running. And at the end, you will have a notification that will tell you that your method terminated correctly. And you will be able to find the output here, as well as in the output path, as well in the CCP execution path if you choose the first two options.
Thank you very much. And so tomorrow we'll show you how you can generate inputs for cephalopods from various sources.
Yes, indeed. Yes, tomorrow we have another. It's going to be also a very good session. Well, thank you all very much, Jean-Olivier and the entire team. Again, as mentioned, for participants, these are the experts that are going to be supporting you. So it's really important to, and this is another opportunity. We have their bios, of course, etc. They're all online. But this is giving you a better opportunity to really see their expertise, to see what they can support you with. So it's good to keep notes on these things. And without further ado, as they say, we're going to pass to Paul Weerheim from Aris. He's going to give us a presentation on DD&AS, I believe. Thank you so much, Paul. It's always, this is another very big subject and will be super useful.
All right, you should be able to see my screen now. Yeah.
Yes, we are.
Yes. I'll give you a brief overview of one of the parts of the Blue Cloud. Blue Cloud also has a data discovery and access service, which was part of the Blue Cloud since the beginning. Yeah. First, I'm going to talk about what is in the data discovery and access service. In the data discovery and access service, we have metadata describing all the data sets of 13 partners from Blue Cloud. We call them Blue Data Infrastructures, or BDIs for short. This metadata was harvested, is being harvested every week from their services, and we publish them again at 2 levels in the Blue Cloud Data Discovery and Access service. And here you can browse through them, view them, and of course order them for download. There are 2 levels to this. First level is the collection level. Basically, we try to aggregate the data into existing collections or themes within the data if there are no collections. Sometimes there are not really easy to define, so then level 1 and level 2 are the same. But level 2 is the individual data set level. So level 1 can contain one collection of maybe 100 of data sets. Level 2 are those individual data sets. You can also order those data sets from level 2. The data discovery and access service first started out as a service to mainly retrieve the data for the Blue Cloud users, but it has evolved into a tool to advise partners within Blue Cloud to expand their metadata if there are holes or inconsistencies, and we advise them to use RDF, linked data, etc. to publish their metadata. So the role has a bit shifted from being a service for the users than more being a service for the providers. How can you reach the data discovery and access service? Well, you can go to bluecloud.org. Under the Blue Cloud platform, it still says services here on the slide. You can find a little page called Data Discovery and Access. You can go to that page and click the buttons to follow to the access service itself. Here you can see the access service, the first interface you'll see if you start using it. Yeah, I can see at number 2 and on the form on the left, you can search for a free text or a date or a geographic area. After that, if you press search, you'll find the collections per BDI found. You can also click on this collection results header and you'll find the results for all the BDIs at once. If we go deeper, if you have found those results, you can also view the GML of those records. Of course, this looks very detailed. Not all the partners have this high of detail on the map, but at least you can see the whereabouts of the datasets. And if we go further, we can, for example, search specifically in Argo. We have created several filters for each partner separately. For Argo, you can filter on platform, country codes, and status, and all those kind of parameters that we have harvested from Argo. Sometimes there are more, sometimes there are less search fields. Depends on the partner. At level 2, you can select the datasets that you find, as you can see on the right. This will add them to a basket like you're shopping on a web shop. It kind of is. After you think you have selected all the datasets you want, you can click on the basket, fill in your order name, and then order the data. This will instruct the cloud. We have a cloud program to fetch all the data from all the BDIs. You can fetch them from 10 partners at once if you want, and it will put them in one big archive. And after the archive is ready, you will be notified, and you can you have two options with it. You can find them under standing orders under your user menu. You can first view view the order by pressing the eye icon. You can view if all the records were able to be downloaded, because sometimes the partner can have an error or the record is not able to be downloaded, or for some other reason. You can see it there, but most of the time all the records can be found. Then we have two options. You can download it, of course, but you can also move it to your data space by pressing the upload icon. If you press the upload icon, you will be asked to press one more button, push order to the VRE, and then you get a confirmation, hey, it will be pushed to your workspace. After that, you can find the order data in your workspace, as you can see in step number 3. Yeah, and that's the basic functionality of the data discovery and access service. To be fair, my colleague has way surpassed me in this year in the start of Blue Cloud. It was very useful, and now we have Beacon, which Robin will tell you about in detail. It's a very powerful tool, and I really recommend you use that. But of course, feel free to use the data discovery and access service however you like. Thank you.
Thank you very much, Paul. And I can tell that you're both fans of each other's work, you and Robin. So it's always nice to see that in a team. So we really look forward now to going to the presentation of Robin Kooyman. Hello, Robin.
Hi, thanks, Paul, as well.
Thank you for joining us.
No problem, no problem.
Let's see if I can share my screen. All right. Hopefully everyone can see the screen.
Yes, absolutely.
Okay, perfect. So yes, hi, I'm Robin. I'll be talking a little bit about Beacon, which is the high-performance subsetting data lake that we've been working on for the past few years within various European projects. So a little bit about the origin of Beacon. A couple of years ago, when I started at Maris, we had millions and millions of NetCF files, and we wanted to provide subsetting access to these millions of NetCF files within minutes of getting your data back to you. So how do you go about building a software that allows users to build a query, subset out of these millions of NetCF files, create one harmonized output, and then they can directly use in their Jupyter notebooks or in some sort of other application. So one of these example requests might be give me all of the temperature data in the North Sea between 2000 and 2010, in degrees Celsius between the depth of 0 and 5 meters. That's the kind of query that we wanted to be able to send to the CDataNet CDI system, and then get back one NetCDF file that would return all of your data in a single file harmonized, and that you could directly use in your notebook to do your research on. This is how Beacon came to be alive. And Beacon is the tool that we use to allow users to have really quick access to incredibly large amounts of climate data that they can use directly. So Beacon in a nutshell, it was written in, or it is written in Rust and C. It's high-performance data lake. It runs on any platform using Docker containers. Consists basically of a RESTful API, basically any standard API. It provides some core libraries, which does all of the heavy lifting, and it provides these real-time subsetting access to really large climatologies. It can produce different output formats, such as NetCDF, CSV, Arrow, Parquet, GeoParquet. A bunch of these formats that my colleagues in the Workbench have already shown and talked about. So Beacon can handle any kind of NetCDF structure, it can handle any kind of Parquet file, and it supports basically any environment it works on, and it allows for really powerful query capabilities and allows for filter on ranges, using these polygons, filter on metadata, using union or aggregation queries, or even federated queries. So what powers Beacon internally is the Beacon binary format. Think of it as a zip file where you put in 1 million NetCDF files and magically it becomes super fast. This is done because the Beacon binary format is what we call fully parallel, which means all of your cores in your system can use it and read and traverse the data itself. And all of this data is then stored in what's called Apache Arrow arrays, which allows for this really quick computational access to this data. And then Beacon binary format also uses what's called serialization calls, which means the file is being read as if it was already in the memory of the computer system, which means it's really, really quick for the computer processor to process the data to the point where you actually only have the data that you're after. So something a little bit about performance is that, for example, we have within the CDI, we have 2.5 million NetCDF file datasets, which contains way over 4 billion actual data points. It's around 200 gigabytes of NetCDF data. And one of the queries might be, let's say I want the longitude between -8 and 12, the latitude between 50 and 61, and the depth between 0 and 50, and the time between 2010 and 2012. And then I want all of these different CDataNet temperature parameters aggregated and harmonized in a single unit in Celsius. And then within 2.5 seconds, I will have a result file, for example, a Parquet file containing 12 million data points, all in unit degrees Celsius, directly usable in your notebook. Beacon can run on any system, and it scales incredibly well with your data. It allows for compressing, so it's really meant to be used by anyone. So one of the things you can do, for example, is you can read through these giant climate collections. For example, this is a slide, or this is a plot that I took from one of the notebooks sometimes I use for testing, where I subset out of the World Ocean Database, the Cora collection, and Argo, and then I plot the temperature for the North Sea between 1950 and 2020 within just 30 seconds of running the notebook. So it's really, really quick. Other examples you might use it on is, for example, the Argo collection. We also have some instances running Arrow 5. The Cora TS example that you can plot somewhere on a map as well. And within the Beacon Blue Cloud, we have various instances available. For example, the EuroArgo, we have Cora Profile, Cora Time Series, Siemens BGC, we have Immunet Chemistry, we have the World Ocean Database, and of course we have CDataNet. All of these are available for everyone within the hackathon to use within the VRE. So how do you go about using the Beacon data lake in the VRE? So first, you install the Beacon Python package, which creates just a better user experience for querying the Beacon instance, because it creates these simple functions to use. Of course, we also have some documentation available that you can follow along, so you can know more about how it works or how to adjust it. And every example notebook that my colleagues have already showed on the GitHub will install this package in one of the first cells. So everybody has this package installed and is ready to be used. So you start off by installing the package, and then you import the library, the Beacon Python library itself. Then you have to actually connect to one of these Beacon data lake instances, one of these Argo or World Ocean database, whatever you want to use. And you do it by first fetching your D4Science user token, and then you fill in the URL of the Beacon instance, which in the case displaying on the screen right here is the World Ocean database instance. And you just run the cell, and in the VRE, it will automatically get your user token, and then it will also, when you see below, set up the connection to the Beacon data lake, it will also connect to the Beacon data lake instance. And underneath, you'll see a message that says connected to blah, 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 blah instance server successfully. And then you know that you're successfully connected, and you can start using the data lake and explore all of its contents. So exploring the contents goes as follows. You can list all of the available columns. These can all be later be used when you're actually querying what's inside of the Beacon. So there are tons and tons of columns available for every different kind of Beacon data lakes. Or for example, the World Ocean database has different columns than, for example, the Argo collection. So feel free to explore what's inside of these data lakes as in terms of contents and available columns. And all of these columns can be used in your query. So don't be afraid to query them. So how do you go about building your query? So a query consists of three parts. The columns to select, for example, temperature, longitude, latitude, time, any other metadata columns, station, platform, instrument. Then you can start to apply some filters on the selected columns, some range filters with some equal or not equal filters. So I want the station to be equal to blah, 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 blah station. Some null or not null filters. You can also create these complex or filters or end filters, and of course the polygon filter. And then you start selecting the output format, for example, Parquet, GeoParquet, Arrow, NetCDF, ODV, CSV, ZAR, any kind of formats that you're after that you can use in your notebook. So you start off by first initializing the query builder. You do that by calling the query function on the client that will return you a query builder, and then beneath you'll see the start adding interest columns. That's where you can start adding all of your columns you're interested in. For example, we see the WD unique cast, the platform, the institute, temperature, WD flag, WD profile flag. And you can just keep on adding more and more columns you're interested in. Then you can start adding some filters to it. For example, we add a range filter for the time to be between 2000 and 2015. And then the longitude, and then we add a filter on the latitude, or even a polygon filter if you're interested in more specific areas and have to filter out certain curvatures. You can also add some equals or not equal filters. So for example, we add an equal filters on the temperature quality flag, which means I only want the data where the temperature quality flag equals 0, which in the World Ocean database means it's good data. And also you can add some is null or not null filters to filter out any missing data that is null because there's no data for it. Then we go to executing your query. Any methods that starts with the to and then something afterwards starts executing your query. For example, the to pandas data frames will actually start executing your query. You'll see that down below. You see the query builder.to pandas data frames will actually execute your query and return a pandas data frame that you can then directly use in your notebook in the VRE. And for more advanced queries, we have some notebooks available on the GitHub that my colleagues talked about. We have some simple variants and some more advanced variants, so feel free to play around with. And of course, we have the documentation available as well. So here you can see the link to the Hackathon GitHub, where you can find the notebooks. All the notebooks can directly run in a VRE, and if you prefer to run them locally on your own machine, that's possible as well. It just takes you another step. Just read through the Jupyter notebook, and you'll see what you have to do to run it locally. If you wish to run it locally, for example, in your VS Code or your own Jupyter, whatever you want. Some examples we have for Argo, Cora, Unit Chemistry, CDataNet, and World Ocean Database. And they're just examples, so feel free to play around with it, add some columns, change some filters, whatever you like. So now we're going into a live demo. We're going to be subsetting through the World Ocean Database, which contains around 20 million NetCF datasets, and we're going to do that within a couple of seconds. So I'll be sharing a different screen. Let's go here. This one. I hope everyone can see the screen.
Yes.
Okay, so here we have the GitHub with all the notebooks. I just grabbed the WOD, World Ocean Database simple notebook, and dragged it into my VRE environment. So here we open the notebook, and we see we're in the World Ocean Database simple. And what I'm gonna do is I'm gonna run the notebook, and while it's running, I'll be talking you through the notebook, and likely it will be faster than before I'm done talking about it. So let's start it, and it's going to run the cells. So first we start off with installing the Beacon Python package and then importing the Python package into our VRE. Then it's going to fetch our D4Science token, which allows us to log into the Beacon instance. And then we add the Beacon instance URL. So we say we want to connect to the Beacon World Ocean Database. Then we're going to connect to the Beacon instance URL, and then we see, hey, we're connected successfully to the Beacon World Ocean Database. Then we can start listing some columns that are available within the World Ocean Database. And here I just search for WMO. You can also remove this, and then you just run the cell again. And then what it will do is it will fetch the list of the entire collection of columns that are available. Of course, because I'm running the notebook, the other cells have to run first. So while I was talking about it, I did a subset for the Mediterranean between 2014 and 2015 for between 0 and 150 meters of depth. And within a couple of seconds, it returned me 1 million rows containing all of your data you're after. So here you see the WT Unicast platform, all of this data. You can directly use in your notebook. And we can see it already plotted the plot for us on the map, so we can see it interactively, or we can see it in our notebook ready to be used for both temperature and salinity. And of course, you can push this further. So for example, we also have a variant. If I can go up, we have the simple global variant where you can query it as well. And what it does is fetch 6 million rows for a year of data for the entire globe, and then you can get these kind of images directly in your notebook. And you don't have to worry about any performance, so it will be really quick. Of course, we have these more advanced notebooks, for example, World Ocean Database, the non-simple version. And what it will do is it will do much more, it will have much more detail in what it actually is after. For example, when we're running to where we build our query, we can see that we can select different formats. So for example, the format is by default, it's on pandas, but of course, if we change it to ODV, what it will do is we'll fetch, or it will run the query, and then we'll output your query as an ODV file instead of it being a pandas data frame or a packet file. So you can change this freely and then run the query, and it will output it for you. Of course, you can add different filters, so feel free to play around with any of the notebooks. And when you run these notebooks, you can also get these diagnostics, as Simona already talked about. So you can see this all in real life appearing for your screen. So that's a bit of the demo. And then I can go back. Yes. Okay, so we're just on that. So if you have any feedback or issues, let us know through Discord, create an issue on the GitHub. And if you have started documentation for the Beacon Data Lake, it's linked as well. And the Beacon technology is also fully open source. So if you're interested in viewing what's actually powering Beacon, then feel free to also check out any of that. And that's it for me.
Okay, thank you very much, Robin. Thank you for this. Both the data and discovery access service as well as Beacon are going to be described in the hack pack that we're preparing and will be launched by the end of this week. But again, I think it's good for everyone to see these presentations live I think it makes a bit of a difference for those who prefer, you know, watching a live demo than just reading instructions. So we are still doing quite well on time. And now we're going to pass to our colleague Sebastian Miaruch from the Alfred Wegener Institute, who is going to tell us all about Web ODV. Thank you, Sebastian, and over to you.
Okay, thank you very much. Okay, great. I will share my browser. So.
Yep, yes.
So can you see it? Yeah.
Yes.
Great. Okay, I'm going to present Web ODV. Now you have heard a lot about it, and now let's see what you can do with it. And I will really start here from scratch, so you can really see how you can use Web ODV in the Blue Cloud VRE. Okay, let's sign in to the Blue Cloud VRE. I'm going to use my Google account, and okay, I already logged in a couple of minutes ago, so I'm already in, but the process should be very clear. Okay, let's go to the Blue Cloud Lab. And first of all, or let's say here on the menu bar, you can see the different apps. We have the Web ODV, which is here. But first of all, let's start a Jupyter Notebook and just look into our workspace. So here we are. This is my workspace, and this looks similar in your case, except that you probably will not find any data if you log in for the first time. So what is crucial is that Web ODV is only connected to your workspace. So this is the only place where you can work with Web ODV. This is important, which means all the data that you are getting out of the system, for example, with Beacon or with the data discovery service, you have to put your data into your workspace, the home directory, which is essentially here in the Jupyter Notebook, it's /home/Jovian. So this is your home directory. And if you want to work with Web ODV, put your data into the home directory. So what I did, I put some data here into a folder, which I called Hackathon. So I have here ODV data, and it's again important to know that ODV data always consists of a .odv file and a .data. So, and this is called an ODV collection, and you always need a .odv and a .data. But ODV will produce that, and I will show you that now in the following steps. Okay, let's start a Web ODV. It's starting up here in another tab, and now this is the homepage of Web ODV, and on the right side you can see the available ODV data in your workspace. So workspace means, let's go back to the Jupyter. As I said, it's really important. Workspace means your home directory. So this is the home directory, and Web ODV can see only the ODV data here that you have in your home directory. But the crucial point is that with Web ODV, you can also import data which have been retrieved by Beacon. And Catalina and Simona already have shown that Beacon can create this ODV output. These are TXT files, text files. And I want to show you one of these files. And as an example, I have here a time series TXT produced by Beacon. And this is how it looks like. Normally, you don't need to look into it. Okay, now it's bit difficult to show it here, but it's just a TXT file, a column-oriented file with all the data in it, and normally you don't want to look into it, but just for your information. So we want to import this data now in WebODV to work with it, and therefore, sorry. I first of all, I want to show you an important point. This is the documentation. So please go to the documentation in WebODV, and what I really recommend are the videos. So we have a couple of videos here. They are not long. They are around 5 to 10 minutes, and they cover the most important points, like importing data, working with a map, creating different plots like surface plots, and exporting data. Also that. So I really recommend the videos, because then you really see where you have to click on and what you have to do. In addition, we have more documentation here, also the big ocean data view documentation, which can be useful if you are searching for a specific topic. And WebODV is based on Ocean Data View. So it's also a good idea to visit the original Ocean Data View website to get some information. So very important, have a look at the documentation. It's very useful. Now let's go to the import service. So the import service consists of a couple of steps. First of all, you have to select the type of import data. In this case, if it's a TXT beacon output, it's a spreadsheet, just a column-oriented TXT file. Then you have to select the data. And here again, please ignore the data space. You are working in the workspace. The workspace is your private workspace under Home Jovian. So Home Jovian equal workspace. Click on refresh to see all the files that you have in your workspace. And here I can locate the time series.txt. So I check it because I want to import it into WebODV. I go to start import, and now, very important, please be careful now, select the workspace you want to import the data into. I mean, this is the output folder for the final ODV data. Please select workspace. Again, workspace, right? This is the space you're working in. And now, just give it a name. I just use the Hackathon folder, the import folder, and I name it time series. This is the name. And now I can click on Start Import. I'm redirected to the first page of the import service, and I see that there has been an import started right now. The output folder is the workspace, and this is a file name or the path of the data. And now I see that the import is running, and I can click on refresh to see if it's already finished or still running, and I see it's finished. So let's go to the home page again. And now we cannot yet see the final data, and I have to click on refresh. Also important, always click on refresh to see if we have new data in the workspace, and indeed we have. There's now this folder import, and WebODV can see now a newly created file, a time series.odv. Let's check back here in the Jupyter. In this import folder, I had this time series txt. Now I have a time series ODV and .data. So, and this is the ODV collection that I need. It's visible now in WebODV. I can click on it. On the next page, I have to click on WebODV Explore, and WebODV will open the data. And now I have the data here in WebODV, and I can work with the data. And as you have already seen, WebODV is an interactive tool. So It's operated with a mouse mainly, and I have a lot of options. And again, have a look at the documentation. You will find videos how to work with ODV. So, for example, I can create a new data window where I have the time on the x-axis, and we are working here with time series. On the y-axis, I can put, for example, oxygen measurements. And now what you see here are the oxygen measurements. And as Catalina, for example, explained, ODV provides quality flags for every sample that we see here. If I click into the data here at the bottom, we see that the oxygen data that is now on the y-axis, we see here in this window on the right, we see the data value and we see the quality flag. In this case, it's 2. If I click on data here on the top, which are somehow looking like outliers, we see it's a 4. And 4 is bad data. So let's do a bit more with quality flags. So you are able to give quality flags to data. For example, here we have a point which is somehow maybe a bit weird and outside. I don't know. I'm not a oxygen expert, but it's probably a bit too much here. If I look on it, it's a 1, the quality flag. If I click here the right mouse button, I can assign a quality flag to that single sample. And here I have a list of all the quality flags, good value, probably good, bad, and so on. And in this case, I would give it a bad value. So I click on apply, and then I see that this sample now has a quality flag number 4 and is now considered as bad. So what can I do now with the quality flags? I can filter the data. What I can do is I can right click, go to sample filter and say reject outliers. And now all the bad data are gone. And I now only have the good data. And ODV provides so much more, which you can learn about in the videos or the documentation. For example, we can go to statistics, curve fitting. Let's say we want to plot a moving average, filter through it. Here we are. There's a moving average filter and much more. Okay. Good. I want to show you, I have a bit of time left. I want to show you 2 more visualization types, and therefore I open one of the data sets here from the Argo dataset. I open it with WebODV, and we see here the Mediterranean. And I just want to show you some nice features that ODV provides. For example, it's a very strong filtering options. One filtering option I like a lot is the polygon filter. So you can define a polygon. Let's say I am interested in a certain region here in the Mediterranean. I can easily click here or create a polygon and select the data visually and very, very fast. And what I can do now, for example, is creating a surface plot. Therefore, I go to one surface window. On the z-axis, which is here color-coded, I can put the temperature. I can zoom into the region I'm interested in. And now I can see all the temperature data here on the surface. I can now, for example, interpolate the data. And I also can add contour lines. Now, I mean, it's just maybe a bit too much contour lines, but you know how this can be done. So this is a surface plot. Another interesting plot is a section plot, which is doable with WebODV. And especially if we have profile data here. So what we can do is we can define a section, and again, this is a manual process, and it's fantastic. I can create fully custom paths here on the map by clicking along a track. And then I have this section, which is in this case maybe a bit too large. So again, we make it a bit narrower. So now this is a section, and now only data within this section are shown. On the Y, we have the depths along this section track, and on the X-axis, there's the distance. We can also change the X-axis and choose, for example, longitude. And now on the Z-axis, color-coded, we can put, for example, the temperature. And now we can see the temperature distribution here. Again, we can interpolate the data. Now we have a nice interpolation. We can add contour lines. So you see, it's a very powerful tool for doing these analysis. Okay, I think that's all I wanted to show, and happy to get some questions. And during the hackathon, please contact me if you have any questions. I'm happy to help.
Thank you very much, Sebastian. And I actually do hope it's something that we keep repeating. We do hope that participants will be reaching out to you. I think that out of everything that we have presented, and we still have to present in the next session, it is the experts behind all of these tools that are really the best resource that all of you can have. So I believe that it's 4:40. We are still quite on time. We can go to Q&A. So please, please open your cameras once again. We had a few questions that we answered in the chat during the sessions. So now we're going to wait just a little bit to see if we do get any more questions. Is there anyone who would like to make any further comments from our guest speakers today? Again, you gave us really great presentations. Thank you. It will soon be uploaded, the entire video, along with some of the resources you have provided will be uploaded on Discord in the coming hours by Michele. Okay. Well, as always, if we do not have any further questions to reply to, we're not going to keep you any longer. Once again, a big thank you to all of our speakers, and I'm going to pass to Michele to give us the next steps and let us know what our next and final session is going to be. Thank you, and I will see you all very, very soon. Michele,
Thank you
over
so
to you.
much. Thank you, Angeliki. So we do have a question, but it's not for the speakers, and it is how many members can we have in our teams? So by terms and condition, you should have at least 3 members and maximum 10 members. So this is the reply. Someone is just thanking us and all the speakers in the Q&A section. So no, thanks to you guys for being here. As Angeliki said, we will record this. So let me share with you, without further ado, the next step. So live session number 4 will be tomorrow. It will be all about the challenges. Many of the speakers, you already met them either today or during the other live sessions. So basically, it's very simple. Challenge 1, 2, 3, and 4, and you will have the chance to ask your questions to these people who are basically the challenge leader. So as you know, we have appointed 4 challenge leaders, Jean-Olivier, Cyrielle, Simona, and Julia Vera. Just to mention that for challenge number 1, there will be some other mentors with Jean-Olivier to help him reply to your questions. So that is it. During live session number 4 tomorrow, we will give you the details for the next steps. We will kick off the hackathon on Monday at 10, and we'll give you all the details to join. And as always, you can then, if you have other questions, you can go to Discord and you know where to find me and where to find us. Just a note that the channels to reach out the panelists here who are also coaches and mentors will be open in the next few days. So they will not be open right now. We will do it right before the hackathon starts. So you can start asking your questions and receive support from them. So yeah, that is all also from my side. Angeliki, if you don't have any final words,
No, that's it. Thank you very much. We will be seeing you tomorrow when we will also be joined. So it will be a joint session between Blue Cloud 2026 experts as well as Digital Twin Ocean experts. So it will be another great opportunity for you to take a look at how things will be working, especially everyone who will be in challenge 4. But I think it's very important that you participate and get a little bit more information on the challenges themselves, on the status quo currently, what Europe is working on, etc., what the world is working on, and what we are looking forward to seeing. Thank you. Have a great afternoon or morning, wherever you are, and we'll see you tomorrow. Bye.
Thank you all. Bye-bye.